Hi, my name is Graham Murray, an MD PhD student from the Reed Lab at Virginia Commonwealth University, and today I'll be talking to you about the spectrum of single-cell drug response through the lens of long-term high-speed live cell interferometry. So an overview of what I'll be talking about today: what exactly is high-speed live cell interferometry, typical drug screen that we use high-speed live cell interferometry for, the spectrum of drug response from apoptosis to growth recovery, and then just future directions. So what is high-speed live cell interferometry? So HSLCI was developed as a clinical tool to predict the in vivo drug response in patients based on the single-cell biomass growth rates of a tumor. What it's made up of is a quadriwave lateral searing interferometer incorporated into a custom scanning system. So what we have here first is an LED light over an objective with a micropipette that's used to isolate cells of interest after an experiment. So these are the two things that are above a motorized stage that scans above the objective, which can be 10 to 20 or 40x. And then as it's scanning, there is an autofocus system so that the distance between the objective and the plate remains the same and the cells of interest remain in focus. And then finally, our quadriwave lateral searing interferometer is incorporated and captures images here. So again, what we're doing is looking at single-cell biomass growth rates. So why do we care about cell mass? So cell growth and proliferation are tightly regulated processes integral to cell functions. There are cells like a red blood cell, which is only 5 microns in diameter, and there are cells like an ovum, which is 100 microns in diameter. Their size, growth, and proliferation are all very dependent for their function. The red blood cells need to fit through your veins and arteries, while the ovum must have enough nutrients to grow an embryo. And growth, proliferation, and death all involve changes in cell mass. And when cell mass becomes dysregulated, disease can result. What we've focused on in particular is cancer, of course, as cells continue proliferating and growing in size despite drug treatment, but also myopathies. If your heart cells get too large, then they no longer function correctly. So cell mass is an integral measurement for many forms of biology. So what does a typical experiment look like? So our in vitro biomass growth experiments typically start with a tumor that's been excised from a patient-derived xenograft. So what this is a tumor is taken from a patient and then implanted into an immunodeficient mouse where it's allowed to grow there. And this maintains histologic and genetic characteristics by growing it in an in vivo setting. So these tumors are excised and then disaggregated into single cells. These cells are then plated on a 24 or 6-well plate and dosed with whatever drug we are screening at the time. In the case of the example I'm showing you, we are dosing carboplatin from 0 to 1 millimolar on triple-negative breast cancer tumors. We then monitor the biomass growth rates of the cells about 24 to 36 hours after treatment. We then compare this to in vivo treatments. So in this case, the tumors are seeded. They're allowed 0 to 24 days to grow. They're given a single dose of carboplatin, and then tumor size and progression is monitored up to about a month and a half afterwards. So these are what our results look like. We have 3 triple-negative breast cancer tumors, HCIO9, HCIO1, and UCD52. To look closer at what one of these graphs is really showing, on the x-axis, we have a dosing carboplatin ranging from 0 to 1 millimolar. On the y-axis, we have hourly growth rate. And then each dot in these box plots represents the measurement of a single cell over the 24 to 36-hour period after treatment began. So as you can see from these plots, HCIO9 was the most resistant tumor, while UCD52 was the most sensitive. We then performed each experiment 3 times to confirm and develop dose-response curves, which again, so HCI09 was the most resistant, UCD52 was the most sensitive. Then we compared our in vitro results to in vivo results. And as you can see here, HCI09 tumors continued growing despite drug treatment at about day 24. HCI01 tumors had decreased growth in comparison to control tumors in black, while UCD52 tumors actually began to regress after only a single dose of carboplatin. So our in vivo results matched our in vitro results. But what's most interesting is actually the sensitive tumor, UCD52. So at a dose of 100 micromolar, which is above the maximum concentration of carboplatin in a patient's blood, you have cells that are dying, but also cells that continue growing despite the carboplatin treatment. And so you can see here's an example of a cell that continued growing despite treatment, while here's an example of a cell dying. And it's these cells that are growing despite treatment that is really key to HSLCI's single cell sensitivity. And can we isolate these cells and learn more about potential resistance forming? But the first step of this is actually exploring what's happening to these cells, not just 36 hours after treatment, but 72 or even a couple days later. And are these cells truly resistant? So over longer time periods, how do the ranges of responses that we see 24 to 36 hours after treatment develop? And which responses are significant and could be sources of drug resistance? So, of course, continuing to grow despite drug treatment is an obvious sign of drug resistance, but is there some subtlety to what's going on, and which cells will continue to survive even days and weeks later? As this is a vast space, because using different drugs will result in different responses and different cells will result in different responses as well, we just wanted to open a window into what's going on here. And the first thing we wanted to look at was cell death. To explore apoptosis and cell death, we looked at 3 cancer cell lines: a lung, kidney, and breast cancer line. We treated each line with actinomycin D and camptothecin to induce apoptosis. We then correlated biomass signatures, which you can see on top, with a fluorescence signal of propydium iodide. So when the cell membrane becomes permeable during the process of apoptosis, propydium iodide is allowed into the cell and then intercalates with DNA and fluoresces. And with variations between the 3 different cell lines, you can see apoptosis happens relatively quickly. And with treatment beginning at 60 minutes, indicated by these blue lines, A549 has lost almost all its mass by 150 minutes since the start of monitoring, while the slightly slower MCF7 is still losing mass 400 minutes after the start of monitoring. And you can see there is a correlation between our fluorescent signals and our biomass signals, indicating what we're seeing is apoptosis. So what kind of implications does this have for drug screening? So recall our most interesting experiment with UCD52, where we had our resistant cells growing despite drug treatment. So typically we're performing these screens 24 hours after drug treatment. But you'll notice that apoptosis is occurring within 5 hours, even in the slowest cell line. So the implications this have is that while these resistant cells may appear to be 16% of the population measured, because with our methods we throw out debris, because if you measured every cellular debris, your median cell growth rate would be 0. Many of the cells that have gone through apoptosis have probably already turned into debris at the time we're looking at. Therefore, these drug-resistant cells are most likely rarer than they actually appear. So what did we want to look at next? Next, we looked at senescence. Cellular senescence is a condition with characteristic changes in metabolism, cell size, which makes it an obvious target for using HSLCI, genetic and epigenetic expression. So classically, these cells enlarge and quit dividing, and it can be induced by DNA-damaging therapies such as etoposide. To explore this, we treated H460 lung cancer cell line with etoposide for 24 hours, removed the etoposide, and then monitored up to 9 days later using HSLCI. Classic senescent behavior can be seen in the panels on the right. So on top, we have the biomass growth rate of the cluster that's shown in the bottom. So you can see we start out with 3 relatively small and more dense cells that over the course of 6 days expand in size without dividing. Typically, H460 cells would have gone through around 6 rounds of division by the time you get to 6 days. So what did this look like at a population level? So we saw a similar trend. So vehicle cells grew exponentially at about 3.58%. And you can see on these graphs here, we've essentially plotted 709 biomass growth tracks and then found the median, which was the 3.58%. And then you can see in B, we have our treatment tracks, where we treated for 24 hours and then monitored up to 5 days later. Vehicle cells could only be monitored for 2 days because they had grown so much they had overwhelmed the plate. The treated cells showed the characteristics like you saw on the last slide, where they grew at about 1.29%, most of them only growing in size and not in number. But there are rare exceptions that began dividing as well. And to explore these rare exceptions, we actually performed flow cytometry for the beta-galactosidase metabolite C12FDG. Beta-galactosidase is typically upregulated in senescence. So after performing cell sorting at day 3, we looked at those that showed C12FDG high fluorescence, so a high presence of senescence beta-galactosidase, and those with SOTA low presence. So those with SOTA low presence had regained the growth of vehicle cells by day 4 after treatment. In contrast, the C12FDG high cells continued as a median growing at 1.29%, but you can see there is a vast change in outcomes by day 7 or 8. There are some cells that grow robustly and others that tend to peter out. And it's the divergence of outcomes in these C12FDG high cells that is most interesting. And it's really those that escape from senescence and continue growing that could be an issue. So you can see here we have an example where large cells begin dividing, and then eventually through generations, they begin to shrink and take on the characteristics of control cells. And this recovery from senescence could actually lead to cancer reoccurrence and could be a source of it. And that's why it's so interesting. So what would we like to do next? We'd like to continue to correlate the behaviors we see on day 2 in our cells of interest with behaviors later down the road, say day 9 or potentially even further, to characterize potential forms of drug resistance and recurrence. But we also want to isolate these cells of interest that we identify at day 2 for early molecular characterizations of drug-tolerant cells. And as you'll see in the video I'm about to play for you, we've incorporated a micropipette isolation system so that we can isolate these cells. A cell of interest is identified by the computer. The user then selects the cell, and a micropipette comes down and isolates it. You can see further characterization of this system in a paper recently released in Analyst under Murray et al. We'll continue to perform drug screenings, like I mentioned in the beginning of the paper, correlating in vitro and in vivo measurements, and also explore other combinations of drugs and the states of drug response, as this is a vast space. I'd like to thank all the people who made this work possible, especially in the Reed Lab, of course, Dr. Jason Reed, Kevin Leslie, and Daniel Guest. Also our collaborators, especially Dr. Gewurtz and Dr. Sayel, who helped with the senescence studies. And in the Harrell Lab, Dr. Harrell and Dr. Turner, who helped with the patient-derived xenograft studies. Thank you.